And a very warm welcome to today's Nucleic Acid Insights webinar, titled Enhancing Manufacturing Efficiency and Quality Through Optimized Lysis and Plasmid DNA Extraction. I'm Lauren Coyle, an editor at BioInsights, and joining me today are Robin Pellizzari and Mateusz Korenc, who will discuss opportunities to optimize scale-up of the pDNA process for mRNA applications. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find more information on the topics covered today. Now, I'd like to introduce you to our presenters. After earning a degree in biotechnology engineering, Robin joined Sanofi as a consultant, leading a project to optimize chromatography and producing thymoglobulin. In 2022, he became a scientist at Sanofi's mRNA Centre of Excellence, created after the COVID-19 pandemic. There, he develops and scales up purification processes for pDNA, contributing to innovations in mRNA technology and bioproduction processes. Mateusz leads analytical support and material development at the Knuth installation at the EMEA region after gaining his PhD in biochemistry. He joined Sartorius BIA Separations in 2021 as a project manager and later focused on inline lysis machine development and GMP implementation. So, without further ado, I'll hand over to Robin to kick off today's presentation.
Hello, everyone. Thank you, Lauren, for the presentation. I'm Robin Pellizzari. I'm scientist in Sanofi mRNA Center of Excellence, and today I will co-present with Mateusz Korenc about the enhancing manufacturing efficiency and quality through the optimized Lysis in plasmid DNA extraction. So first, I will do a plasmid extraction overview on the different way to make this extraction. After, I will more focus on the alkaline lysis method and the challenge for the development of this method. I will talk about the industrialization challenge and the different bottleneck we can have for the pDNA extraction. And after, Mateusz will present you the Sartorius inline lysis system, so the alkalizator, the application of this system, some case study, and we will talk about the evaluation we have done in partnership with Sanofi last year. So, as you know, the plasmid DNA is a crucial component in the production of the next generation therapeutic. So it can be used in direct injection, transfection, in vivo or ex vivo raw material for the viral vectors. But in Sanofi mRNA Center of Excellence, we use it for the RNA manufacturing. So to better understand the application of this plasmid in Sanofi, here is a slide presenting the multiple steps of the process. So first, the plasmid is transformed in the bacteria to generate the cell bank. After, we have the fermentation to generate the product. We have the pDNA purification, linearization of this plasmid, purification of the linear plasmid, and we obtain here the linearized DNA template. So this product is used for generate the drug substance with the in vitro transfection and the capping to generate the mRNA. We have the different step for the mRNA purification, and we obtain here the drug substance. After, we have different step for the formulation, so for the drug product. The mRNA is put in nanoparticle of lipids, so this is a formulation, and we have a purification step for this drug product. After we have obtained the bulk drug product, it's filled and finished. So here we see that the plasmid is an essential element. It's the raw material for the mRNA process. So to generate this plasmid, it's in sufficient quantity and with the required level of quality. It's necessary to have a robust purification process and more specifically, a robust plasmid extraction method because the purification starts here. So the purpose of this extraction is to recover the plasmid by ensuring the quality and the quantity of the product, but also by allowing the best retention of impurity to facilitate the subsequent purification step. So here is a slide to show you the different way for the plasmid extraction. The first one is the boil method. So this method involves heating the cell at 100 degrees to lyse them. So it's a method pretty simple and quick, but more difficult to industrialize because it will be difficult to heat large volume. And also, if we want to work with a single-use process with bag, it will be difficult to heat at high temperature. The second possibility is a sonication. It's also simple and quick. It's a high-frequency sound wave that is used for break the cell. So the disadvantage of this technique will be the plasmid quality that could be impact if we don't use the wrong parameters. And also we can have a low purity because we will extract all what is in the bacteria, so the protein, DNA, and RNA. Another possibility is the enzymatic reaction. So we can obtain a high purity and it's pretty used for small scale. But if we want to use at high scale for industrialization, the enzymatic reaction with lysozyme or protease could be really costlier. The last possibility, and this is the most widely used possibility, is the alkaline lysis. It's really simple and cost effective. The disadvantage will be the intermediate purity. And also we can have a high dilution effect because we will add different buffer, some buffer for the lysis, but also buffer for neutralization. So in Sanofi, we use the alkaline lysis for the extraction of the plasmid. So it's why I will present you more in detail this technique. So here, it's the representation of a bacteria. You can see here the different components that are in the bacteria. You have some protein, RNA, genomic DNA, and the product of interest, the plasmid. So the step number one will be the cell resuspension. So the resuspension is done in the preparative buffer with the EDTA to protect the pDNA from the enzymatic reaction activity like the DNAs, for example. The second step will be the lysis. So we will put the resuspended cell directly in contact with the lysis buffer. So in this lysis buffer, we will have some sodium hydroxide and detergents such as SDS. So this SDS is necessary to make some permeability in the membrane and to allow to the sodium hydroxide to go directly in the bacteria. So the high pH generated by the sodium hydroxide of the lysis solution will generate immediately the denaturation of the plasmid and the gDNA. So the two strands are separate. What is important to have in mind that it's for this lysis, we have to have a contact time that have to be controlled. And this contact time should be between 3 to 5 minutes. After this contact time, we will add another buffer, the neutralization buffer. So this buffer is composed of high potassium acetate concentration. This solution will generate the renaturation of the genomic DNA. Due to this size, it will generate an insoluble network of DNA and it will precipitate. For the plasmid, it's smaller and it will be able to renaturate it and it will keep its solubility in the solution. The high potassium acetate concentration induces the precipitation of the SDS protein complex and also the high molecular weight RNA. The product we obtain here, it's what we call the crude lysate. So to increase the removing of impurity, we will add some salt. It's a precipitation step, and we can use, for example, the calcium chloride for this precipitation. This salt will allow to remove smallest residual RNA. In the same time, it's possible to add filtration head, like, for example, the diatomaceous earth, to absorb the cellular debris, and that could improve the filtration capacity. The last step will be the clarification. All the elements that are precipitated will be eliminated by the clarification on a charged depth filter. So these elements that are RNA, genomic DNA, or protein, or the cellular debris are retained on the filter. The plasmid is not retained on the filter, and it will go through the filter, and we obtain the product that we call the clarified lysate. So As you can see, there are many parameters for the alkaline lysis, and all these parameters could impact the process. So the objective for the lysis will be to maximize the yield and to minimize the residual impurity level in the product. So for this, we have to choose really the key parameters and the good level for these different parameters. The first one will be the concentration of cell after the resuspension. If it's too low, we can have an excessive lysis with damage on the plasmid. If it's too high, we can have an incomplete lysis and so a low pDNA yield for this step. The second point will be the concentration of the lysis buffer. If the sodium hydroxide concentration is too low, we can have a low pDNA extraction, so a low yield, and also higher impurity level, like the gDNA, for example. And if it's too high, we can have some damage on the product. For the SDS concentration, if it's too low, we'll also have an incomplete lysis because we will have a bad permeability of the membrane, so a low yield. And we can also have a low protein precipitation. If it's too high, we can have some solubility issue in the lysis buffer. Another point is the lysis time. If it's too low, we will have an incomplete lysis. If it's too high, we can have some excessive lysis and damage on the plasmid. The mixing will also have an impact. It could be the mixing during the lysis, so when we put in contact the resuspended cell with the lysis buffer, but also when we had the neutralization buffer in the crude lysate. If the mixing is too low, we can have some heterogeneous condition, and during the lysis, it will represent some local extreme pH, so an impact on the quality of the product or the yield. And if it's on the neutralization, we can increase the lysis time, and we can also have an impact on the quality of the product. In any case, if the mixing is too high, we will damage the product. And also, we can have a genomic DNA fragmentation, and this could have an impact on the rest of the process because the fragment of gDNA will be more difficult to remove. The last point that for me could impact the lysis is the precipitation salt concentration. If it's too low, we can have some high residual RNA. If it's too high, we can have some pDNA precipitation and a decrease of the yield. So as you can see, given the complexity of defining the appropriate parameter of alkaline lysis, it could representing a challenge for industrialize this step. So the first thing to have in mind for the scale-up of the lysis is we will have also to increase the quality of the product. For in Sanofi, for the mRNA preclinical tests, so when we work at lab scale in research and development, we can work with a pDNA research grade. When we will go to the pilot scale for the mRNA clinical trial in phase 1 and 2, we can work with a pDNA GMP principal grade. But when we will work for phase 3 or for mRNA commercial at industrial scale, we need to have a pDNA with a GMP compliant grade. So it says that all the steps and all the equipment need to be qualified and validated for the process. Scale-up as well as the quality require many challenges for the lysis step. So here is a slide where you can see the different technical challenge related to the lysis. So the first thing is the increase of the volume. So for the fermentation, we will go from 3 liters to 300 liters for industrial scale. So this increase of the fermentation volume will generate more lysate. So we should have a crude lysate around 1,000 liters. This high volume will generate some difficulty to control the lysis time. It's why it's really necessary to work in continuous mode for the scale-up. To have a good representativity between each scale. The second point will be to assure the product homogenization and to have an efficient mixing system for the industrialization. Another point will be the difficulty to manage the filter head, because with high volume, we can have some difficulty to use this product because it could sediment easily and it's difficult to work with high volume. Another point is the depth filter area. You can see here for industrial scale, we can imagine work with a 30 quarter depth filter. So it will represent high footprint, and also we will need some high volume of water to rinse this filter. The high volume impact will be also the step time that will increase compared to the lab scale. And also, we can have some difficulty in the waste management because, as I said before, we will have some EDTA in the process and it could be difficult to treat. As I presented before, when we will increase the scale of the process, we will expect to have a higher quality in the process. So we have some expectation related to the GMP environment. The first one will be the step automatization to assure the reproducibility between the different batch. The second point will be the process robustness. We need to be able to reproduce the same thing between the different scale, lab scale, pilot scale, or industrial scale. Another point will be the simplification of the Lysis. For this, we need to work with single-use system and equipment to avoid qualification or validation of cleaning. And the last point will be the limitation of the COGS. As you know, when we work in GMP environments, the equipment could be really expensive. So it's why the objective will be to limit this. So to answer to all these requirements, Sartorius has developed an equipment to automatize the alkaline Lysis step. So I leave the floor to Mateusz Korenk that will present you this technology more in detail.
Thank you, Robin. Thank you for this nice introduction. Let me just say that I'm really happy to co-present with you. We are collaborating with Sanofi. Very great, so I'm really happy for this presentation. I'm a product manager for this machine that I'm going to present you right now. It's a Sartorius inline lysis system, also known as an alkalizator. It has a really kind of a strong name. We really love it. It does sound like Terminator, so it makes sense because you are terminating bacteria cells. So let's dive in because the questions were raised by Robin, and those are very valid questions. And I believe I do have answer for all those questions. Now, Alkalizator is a fully automated inline lysis system. It has a tower and a consumable. The consumable is a closed single-use loop, and you can process up to pilot scale. At the moment, it goes up to 500 liters of resuspended cells per workday, and where it really shines is in its state-of-the-art mixing control, which I'm going to talk about a little bit later, why this actually makes a difference. It was tested on many sizes of plasmids, like small 4,000 up to 24,000 base pair. Works very nice. And where it really shines is because it's robust. It actually enables you to have your results more reproducible, and it's also able to scale up and scale down. Okay, now where would you actually find alkalizator? Well, you would find it in the midstream, right? Well, midstream is just between the upstream and the downstream. Midstream is basically the lysis step, right? And I believe lysis step is not talked enough because if the lysis step is bad, you will have a lot of problems on your filtration and on your capture step, and God forbid also in the final formulation. The impurities that can come from a bad lysis can really give you a lot of headaches. I believe the most important impurity is genomic DNA. And if you have a lot of genomic DNA from the start and a very poor lysis step, then you can have like a lot of problems down the line. Usually the midstream is done in batch mode, so batch alkaline lysis. Now, batch alkaline lysis usually looks like on the upper right corner. Now, sometimes it's better, sometimes it's worse, but what you would see is a suspension with flocks all around. Now, alkaline lysis, it's typically done in batch mode. It was first described in the first publication in 1979. So it's already 46 years old and very popular, still very popular in industry. It's actually an industry standard. And it works very nice on a small scale. So in smaller volumes, works very nice. Now, problems start to come when you start to scale up. When you have large volumes, you lose the precise exposure to sodium hydroxide. And if you have large volumes, you also need to mix efficiently, right? And if you are mixing efficiently, you're also inducing shear. And if you have a lot of shear, you can also damage your target pDNA. And if you have also a lot of genomic DNA, then you will shear genomic DNA as well. So the worst case scenario there is that you are going to be shearing your genomic DNA and have these small soluble particles in your filtration, your capture step, and God forbid, in your final formulation. So although batch lysis is very popular, it does have its downside. At the moment, the inline lysis is a perfect alternative. You know, you have this adjustable flow and precise sodium hydroxide exposure to your target pDNA. And here we're also going to be presenting that you have also efficient neutralization. Now, neutralization is something that is not talked enough. What we are seeing in our experiments is poor neutralization usually is seen in more genomic DNA. Basically, the theory goes like this. If you if you are neutralizing slowly, then genomic DNA has more time to become soluble again. Now, if you are neutralizing aggressively and really fast, in a small confined space, then genomic DNA will be captured in other insoluble particles like proteins and RNA and so on. So efficient neutralization is very important and kind of correlates to your genomic DNA presence. Like I already mentioned, the inline lysis will improve robustness, and the inline lysis also improves your scaling up and scaling down. On the picture, on the bottom picture, on the bottom right picture, you see what we were able to do with the alkalizator. We have such better mixing control, such better solution addition that results are very kind of a very comparable here. The only thing that we were actually playing around here is the addition of air. So air here has a direct effect on the flocks that are on top. Now, this is very important to have flocks on top because you can already see where I'm going with this. If you have flocks on top, you can just start pumping from the bottom and have a clear solution from the start. So you're pumping from the bottom, have a clear solution from the start, and prolonging your filter life by a factor of 5 or even more. Now, I'm talking about alkalizator so much that it is good to actually go a little bit into details, how the whole system actually works. So we said that we have a tower. This is the brain of the machine with all the sensors, computer, and so on. And then we have the consumable. The consumable in this case is the lysis reactor. Lysis reactor is the body, the white part, the donut and antenna, plus all the tubing. So we jokingly say always that it does look like an octopus. Yeah. And yeah, it makes sense because you just kind of take this octopus out, put all the tubes on the place of the tower and click play and the machine will do its rest. So what do you have? What do you need to prepare? How do you prepare what do you need to prepare before you do the lysis? You have to prepare bacteria cells that are resuspended. You need to prepare the lysis buffer, which is usually the mixture of sodium hydroxide and some sort of detergent. The neutralization buffer here does not need to be pre-chilled because usually why people are using pre-chilled neutralization buffer is that because neutralization is an exothermic reaction. In this case, we are neutralizing in a very small space. That heat can be really dispersed rapidly. So we can also use a room temperature neutralization buffer, and you also need a precipitation buffer. In our case, this is usually calcium chloride, which effectively precipitates those pesky RNAs and proteins and so on. So how the whole things actually starts is that you first start pumping resuspended bacteria cells and the lysis buffer. These two go at the bottom, and then you start they're mixed at the bottom at the first mixer of the lysis reactor. Then they continue, this lysate then continues to the tube part, which is exactly 1 liter in volume, and then goes up. And right before it meets the neutralization buffer, you see a small yellow dot. This is where aggressive neutralization happens. This is why I'm also mentioning this aggressive neutralization, where you are neutralizing and reducing the genomic DNA being soluble. And then this is also where the flocks are formed. They're pushed to that yellow part. We call this the antenna part, which is a secondary mixer. Here we are adding air. Now, air is added because you're reducing the viscosity, you're increasing the turbulent mixing, and flocks also capture a little bit of that air, making them float like you saw on the picture. The last component that is added into that secondary mixer or that antenna part is the precipitation buffer, or calcium chloride in our case, which efficiently precipitates those impurities. And what you get out is a crude lysate with flocks on top. This is the whole idea. If you have the flocks on top, you can continue pumping from the bottom and have a continuous filtration. And you can also go one step beyond that. If you're using a technology like a monolith, which really is not affected with volumes, you can also do inline dilution. So you can also put the capture step inline. So have everything, lysis, filtration, and capture step inline, which is perfect. So something that also have to keep in mind, we always say to the customers, and this is something that I'm seeing all around the world, if you don't process fast, you will lose your target pDNA. Something to keep in mind for later on. Okay, let's look at some of the case studies, right? Now, these chromatograms were done using our PatFix platform. In this case, it was PatFix pDNA platform. This is an analytical system that we actually developed here at Sartorius BS Separation. Works very nice for really, really dirty samples because the analytical column is a monolith, a very small CMAC pDNA column. So all these chromatograms were done in the same fashion. In this case, what we're seeing in the first case studies, we are comparing batch mode versus inline mode. So the inline is the red one, the green one is the batch mode. And what we can see is that the productivity is very similar. The quality of open circular, linear, and supercooled at the bottom right is also very similar, but we're already seeing that genomic DNA is reduced in this case. So this is already something that I want to point out here, that with inline, you can easily reduce the concentration of genomic DNA. Now, second case here is we actually took crude lysate, filtered it, and then it was analyzed. And fine filtrate after 1 hour showed that we have 7% of open circular and 90% of supercooled. Now this fine filtered lysate was then stored in a refrigerator at 4 degrees Celsius overnight. And what we see is that the open circular concentration actually doubled to 14, only on the expense of supercooled. And even worse, we took the fine filtered lysate and stored it at room temperature overnight, and we see that the open circular concentration tripled to 22 degrees, only on the expense of supercooled. So this goes back to my previous kind of a statement that if you have a plasmid that is really not stable in a crude lysate, then you have to process really fast. You have to go to the capture step as soon as possible. Once it's captured, it's really stable. You can just store it in a refrigerator for weeks and it will not make any changes. But if you store or leave your target pDNA in a crude lysate for too long, you will first see that you get much more open circular, only on the expense of supercooled. So this is something that is, we don't want that. At the very end, you want as much supercooled as possible. Okay, so the first case study, this one is also very interesting. This is more interesting for the research purposes, for people that are more in research and process development, because you can play around with parameters. Because if you have an inline process, you can also play around inline with the parameters. Here we have 6 combinations. So you can imagine the process was running. So the inline process was just running, you are processing, and then you are changing parameters on the fly. In this case, we were changing the parameters highlighted in yellow. So neutralization, precipitation, and air were changed, and the sample was actually taken at the very end. So this whole experiment took something like 10 minutes or 12 minutes, but you got 6 different samples. And you can visually see how your change actually affected the sample, which is great. But you can also put next step or step forward, and you can analyze this. Again, we analyzed all these 6 samples using our CMAC pDNA column and our PathFix platform, and you can see what works best, what doesn't work that good. You know, which you can easily see. You can easily do a screen like on the fly, basically. So usually customers, when they see this, they're like really happy. They're like, yes, this is something that we really want. This is something that is really simple, easy, and very effective. In this case, let's look at number 6. So number 6, let's look at it at the top right corner. You see it's very clear. The flocks are very, very small. So let's see what happened here in the table below. So number 6 had air intake at 5 liters per minute. So in this case, the air intake, so the addition of air, was the highest. Basically what this means, the turbulent mixing in that antenna part, if you remember that I was explaining, the turbulent mixing was so high that we were just kind of shattering those flocks to tiny little flocklets. And they're still floating, which is good. But further analysis is showing that linear pDNA is actually 4%, which is the highest among all. Basically meaning we were too aggressive in this case. We were actually damaging our target pDNA in this case. So it makes sense to kind of reduce the air addition. And let's say in this case, I would say the number 2 would be kind of the way to go here, because you have the best recovery and the best quality. So what am I saying here? It's just that you can easily do a lot of screening. You can easily get a lot of valuable information just from one basic run. So we have the tower. So we have the same tower that can process 2 different lysis reactors. One is smaller one is larger. So the smaller one is 1 liter and the larger one is 5 liters. So basically, if you are having small samples or if you really are not afraid of processing longer, then you can easily go with a smaller lysis reactor. In another case, if you want to process larger volumes, let's say up to 500 liters of resuspended cells, you need to go with a larger lysis reactor because you're going to be pumping faster at higher flow rate, and you can actually process this in 1 workday. In this case, that would be, I think, 7 hours. Okay. Both of these lysis reactors and both towers are in off the shelf as non-GMP and GMP. The tower is also CE compliant, CUL compliant, so it can be also sold in United States, and the software is Part 11 compliant, while on the other case, the lysis reactors can be non-GMP or GMP, and they're already sold internationally all around the world. So I think I actually answered that there is a solution to the questions Robin actually pointed out. So Robin, maybe you can actually now take over the word and show us what can be done.
Thank you, Mateusz. Yeah, in Sanofi, we have evaluated the alkalizator last year. So we have evaluated the 1 liters reactor. And here is a slide with the conclusion we have of this study. The first point is really the process simplification, because we have a removal of the filter head in the process. So it's really more easy to use. Also, we have an increasing of the filtration capacity and feasibility, because we have replaced the depth filter by a frontal filtration thanks to the alkalizator, and we have a reduction of the filter size. You can see here, for example, for the control process, compared to the process with the alkalizator, we have reduced the size of the filter. The second point will be the maintain of the extraction yield from the Lysis. Here, it's the representation of result we have on two pDNA quantity, pDNA 1 and 2. And you can see we have the similar quantity extract between the control process and the process with the alkalizator. Around 200 milligram for the first one and around 300 milligram for the second one. So we assure a good quantity of pDNA content. And the last point that are really impressive for us, it's the increase of the gDNA removal. So as I presented before, the mixing could have some impact of the gDNA fragmentation, and this gDNA will be really more difficult to remove after in the process. So here, with the alkalizator, we have no mixing of the crude lysate, and we avoid this gDNA fragmentation. And you can see that the result we have a better gDNA removal in the clarified lysate with the alkalizator for the 2 pDNA compared to the control process we had before. So really, this is the most important thing we have observed with the system.
Okay, perfect. I think this actually comes to almost to the end almost. These are the conclusions. Well, I think that Robin actually presented very nicely that alkaline lysis is still a way to go. It's still a cost-effective and essential step in the industry if you're in the pDNA world. Again, although it's already a 46-year-old method, it's still very popular. And the only way to kind of optimize it and make it like next level is via inline lysis optimization. You will ensure more reproducible results. You will have more robustness, and you will actually be able to compare batch to batch. If you have a single-use system, this is perfect for the GMP environment because you're then not worried about endotoxins and so on. And alkalizator is the way to go in this case. This is actually a leader on the market at the moment because we do have an off-the-shelf solution, non-GMP and GMP with the state-of-the-art mixing. Something with the scalability, I already kind of teased you at the beginning, but I can already tell you now that we're already kind of looking into the future being Q1 next year, that also smaller lysis reactors and larger lysis reactors will be available. The smallest one will be 40 milliliters. Those are like really perfect for screening or for small scale testing or maybe for a continuous process, while the 25-liter lysis reactor is kind of really a monster. In this case, we're talking about thousands and thousands of liters. Yes, this is it. We're at the very end. Thanks again to Robin and Sanofi team. We have very nice collaboration. So very happy to have this joint presentation. All right. This is it. Questions?
Thank you both for a great presentation there. So yes, as mentioned, we are now moving on to the Q&A portion of the webinar. So please make sure that you are still sending in your questions and we'll get to as many as we can before the end of the webinar. So the first question we have here is, what is the typical yield?
Maybe I can start here. So the typical yield, we have to be careful here when we are interpreting this. So the typical yield usually consists of supercoiled, open circular, and linear pDNA. Now, the best is to have as much supercoiled as possible. And like I already presented, in my case, we are seeing that if you have a small scale batch, which is very efficient in mixing, you are almost one-to-one with the inline lysis. So the yield would be actually very comparable to batch, but once you scale up, the yield with the inline lysis starts to get better. Robin, would you like to comment on that one?
For us, for the yield, it's pretty difficult to estimate because when you want to make the yield, you have to have exact quantity before the lysis and after the lysis. So in the clarified lysate, it's pretty easy to have a quantification. But when you want to make to know the real quantity from the fermentation, you have to extract the plasmid, for example, with the mini preps. So it's pretty difficult to estimate a yield on the process for us. So I don't really have a value. The only thing we have is we have compared the alkalizator to the process we had before, and we had extract the same quantity of product.
Amazing. Thank you both for answering that one. And our next question here is a bit of a two-parter. So the first part of the question is, what size of the pDNA is used for the experiments? How does the size of the plasmid affect the yield? And how does the GC content of the plasmid affect the yields? So sorry, that was a bit of a three-parter question there.
No, it's okay. I understand the question. It's a good one. So the plasmids that were used for experiments in the slides, they're mentioned in the slides. They were smaller ones, like 4.7 kilobase. Those are tend to be like on a smaller scale. The larger one was almost 7,000 base pair. But we do have experience and also data on the larger one, like 24,000. Those really larger plasmids are then used for the AAV production, so helper plasmids and rib-cap plasmids and so on. So we did tests on those. They're actually holding very nicely. It very much depends on the upstream. So the upstream here needs to be very careful to have a good material ready. But yeah, the inline lysis can actually process those, no problem. The GC content, now I can't really answer that one because we were not focusing on the plasmid sequence. So I don't have any comments on that. Robin, maybe you?
For the first question about the size, in Sanofi, we have all the plasmid are similar size. It's pretty small plasmid, under 7 kb. So I don't know if there are some impact for higher plasmid because we don't have tested. And for the, as you, Matthews, for the GC content and the effect of yield, it's not something we have studied. So I don't have some answer about this.
That's no problem at all. Well, thank you both again for answering that one. And the next one we have then is, what factors influence impurities, especially when it comes to endotoxin levels in the process?
Yeah, that one is a very good one. We have to understand that the lysis step is the dirtiest step in the whole process. Here we have like the highest amount of impurities and endotoxins being here the highest. Even measuring endotoxins in fine filtered lysate or crude lysate is almost impossible because the concentration of the endotoxins is just so high. What we did, the measurements that we did here in Sartorius BS separations are showing that concentration of endotoxins in the lysis step, the crude lysis, is very much comparable with the batch mode. So inline and batch were very comparable. It is very important then in later stages, how do you then remove these impurities like endotoxins through filtration and most importantly, capture step, polishing step, and other steps if they're put in the sequence. So I would not focus on the endotoxins in the lysis step, like the crude lysis. Endotoxins are more important in the downstream. Yeah, Robin, would you like to add?
Yeah, I have similar answer than you. For the beginning of the process, there are so much impurity that it's pretty difficult to estimate all this impurity by quantification. So for the endotoxin, for example, with more of quantification at the end of the process, so after the TFF and chromatography step. And the only thing I can say that before the use of the alkalizator or after, we don't see some change on the impurity removing for the endotoxin part, for example. And we are always under the specification, and we have low quantity at the end of the process, so we are good. Really, the only thing we have observed is a decrease of the gDNA level when we have used the alkalizator, because there are an impact about the mixing in the process and this impurity.
Wonderful. Well, thank you both. And the next one, again, this one's a bit of a two-parter. So how can we export data from the machine, and is it possible to customize it to fit specific needs? And is the system flexible enough for different setups?
Okay, yeah, this one is a bit technical, but to export data from a GMP instrument, you need to follow some Part 11 compliance rules. In this case, you can export data via OPC UA platform or via PLC. If somebody from technical side is asking this, he knows what I'm talking about. You can connect your machine to your server and export the data like periodically. Otherwise, the machine does make an audit trail, a detailed audit trail. So after every run, an audit trail is done, packaged, so you actually have something that is from end to end encrypted. Yeah, system is flexible to a certain extent, but there are so many combinations, so many wishes to be compatible with this and that. So this is really like case by case in this case.
Wonderful, thank you so much. And then when it comes to scaling the alkalizator, how do you make sure it works well, whether you're scaling up or down, and what should we be paying attention to?
Okay, another very technical one. Yeah, scaling up and scaling down is it's not trivial. I want to mention this. You have to be really careful when you're scaling up and down. First, because you have to be very careful which parameters are you really looking to scale up and scale down. In this case, the Lysis reactor, I showed the bottom part with the tube and the upper part with the antenna. So the bottom part is really assembled in a way that we have a laminar flow, so we don't have any high shear mixing, so a very laminar flow there. But at the top part, we do want to have a turbulent mixing. So those are parameters that you can then efficiently scale up and scale down. Yeah, in that case, you can go really small. In this case, the smallest one, like I mentioned, is going to be the 40 milliliter Lysis reactor. Those small reactors were already tested, and they show very one-to-one results. So the scaling down and scaling up was already a success.
Well, thank you.
Mm-hmm.
And can the alkalizator be used for other applications, like breaking down different types of cells or capturing molecules other than pDNA?
So yeah, alkalizator at the moment and how it was presented, it's designed to process bacteria cells to extract the pDNA, but we are already working on extracting other molecules. Now, I don't want to go too details in here, but yeah, we know how the mixing works. We know how the lysis works. So we are already kind of working into other fields, like lysing other types of bacteria and also extracting other types of target molecules. So in a nutshell, yes, it can be used for other molecules.
Well, thank you. And what standards do you follow for GMP wetted parts, and how do you ensure they're safe? And further on from that, what's the concentration of endotoxins in these parts?
Oh yeah, endotoxins again. Yeah, endotoxins are important to monitor. You really don't want to have endotoxins in your GMP wetted parts. Now, GMP is a very strict falls under very strict rules. In this case, the wetted parts that I presented, the lysis reactor with all the tubing, is carefully chosen to have high-grade material which can be gamma irradiated. So the whole lysis reactor is assembled in a clean room environment. It's irradiated at approximately 50 kilograms, so it's sterilized, double pouched, and so on. Leachables, extractables, all those tests are done. So we are making sure that nothing goes out, nothing is leaching out.
Fantastic, thank you. Yeah, and I know the lysis reactor is supposed to be single use, but is there any chance it can be reused? And if so, how many times would that be safe?
Yes. Well, it is a single-use component. It is stated as a single-use component, but for research grade, for process development, for everything that is not in production, it can be reused. Now, this is something that it's on the side of the customer to actually decide, but you can just put a cleaning in place, dip it with some sort of sodium hydroxide, wash it thoroughly with water, purge it, and you can use it for another run. Yeah, for research purposes, this can work, but for the GMP environment and production, absolutely no. You need to ensure that you are really as clean as possible, and in that case, only single use works.
Yeah, it's something we do in Sanofi. For the GMP batch, we don't reuse the reactor, but we use the reactor for the GMP team in research grade. So like this, we can reuse it. And for example, we have one since January, and we don't have changed the reactor since. So
Perfect. Perfect. Thank you.
Wonderful. Well, thank you both for answering those questions, but that is all we have time for today. So any questions we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And we'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources tab on the right-hand side of the screen or in the on-demand email. Additionally, an attendance certificate is available on the right-hand side too. So all that's left is to thank Robin and Mateusz once more for a great presentation, and thank you to you, our audience, for listening. We hope you'll join us again soon.
Thank you.